students, and welcome back to KTH e-learning portal. Let me start by introducing myself to you all. I'm Shazeen Khan from Department of Botany, KTHM College, Nashik, and today we are here to discuss the topic of mineral nutrition, which is a topic in the chapter Plant Nutrition for class MSc Part 2, and we'll start with the today's lecture. In the last lecture, we had discussed about soils, the different types of soil, how it is formed, the structure of soil, composition, and the role of soil in detail. So that was the introduction chapter in this credit, which we had started in the last lecture. In today's lecture, we will be seeing about mineral nutrition. Then we will be seeing what are the different types of essential elements that are required for plants. And we will be seeing the role of minerals in plants. Now, to start off with today's lecture, let us get ourselves introduced to the concept of mineral nutrition. Now, green plants, they are autotrophic in the sense that they are independent of outside sources of organic substances. They require inorganic matter from outside and they synthesize their own organic matter. Since the inorganic elements required by the plants, they are chiefly obtained from soil where they are present in the form of minerals. So that nutrition or those elements, they are known as mineral nutrients. And the nutrition which is obtained from these mineral elements, it is known as mineral nutrition. Now these mineral elements or these mineral nutrients which are required for the plants, they are classified into two main categories, which is known as macroelements and microelements. Let us see what are those. Now, macronutrients or macroelements, they are required by the plants in large amounts, which can be at least 1 milligram per gram of dry matter. And the next class or the next category is the micronutrients or microelements. They are also known as trace elements and they are required in comparatively lesser amounts, which can be up to or lesser than 1 milligram per gram of dry matter. Now let us see the differentiation or the distinguishing characters of macro elements and micro elements. You can see the table in this slide given. Now macro elements, they are the nutrient elements present in plants which are available in easily detectable quantities. On the other hand, micro elements, they are present in very minute amounts or they are present in trace amounts. They are present in lesser amounts in comparison to macro elements. The microelements, average amount of microelement, they are present in, which is present in 1 gram of dry matter, it is usually more than 1 milligram. And on the contrary, microelements in 1 gram of dry matter, it is usually lesser than 1 milligram. In case of macroelements, if a slight excess of these elements are present, so they will not be toxic to the plants. They will not have any side effect or they will not have any damaging effect on the plant. But if microelements, they are present in a larger amount or if even slight concentration of these microelements is increased, they can prove toxic to the plants. Now, macroelements, they are generally required to build up the plant body, regulate osmotic concentration, and they take part in biochemical reactions. Microelements, they are required for the functioning of enzymes and they usually act as cofactors in enzymes or they even act as metal activators. Now, to determine which of these elements are essential for the plants, there are some properties. Now, at present, only 16 chemical elements, they are known to be essential for the growth and development of higher plants. In absence of any of these elements, the plants, it develops characteristic deficiency symptoms. Now, to determine which of these element is essential, a criteria was suggested, which is known as Arnon's criteria, and it was suggested by Arnon and Stout in 1939. So there are three principles by which the essentiality of elements it can be determined. Let us see the first criteria. Now, deficiency of a particular element results in the failure of the plant to complete its vegetative or reproductive stage of life cycle. That is, if a particular element is absent from the nutrition, from the complete nutritional requirement, a plant it will fail to complete its vegetative or reproductive stage of life. Let us see the second criteria. Such deficiency symptom, it is characteristic of a particular element and it can be recovered by supplying this element. For example, a particular element, it will lead to a particular deficiency symptom. And that deficiency symptom, it can be recovered if artificially we are supplying the plant with that necessary nutritional element. The third criteria, such an element it is directly involved in the nutrition of the plant. It acts as a constituent of organic molecule or takes part in a biochemical reaction. So these are the three criteria by which the essential elements or the essentiality of elements it can be determined. And on the basis of these criteria only, 16 uh, elements they are considered to be essential for the regular growth and development of plant. You can see that in the slide in the table given. Now the macronutrients which are essential, they are as follows: carbon, oxygen, hydrogen, nitrogen, potassium, calcium, phosphorus, magnesium and sulfur. And micronutrients which are considered to be essential, they are as follows: iron, chlorine, manganese, boron, zinc, copper, and molybdenum. So, based on these three criteria, which is known as Anand's criteria, these 16 elements, macronutrients and micronutrients, they are considered to be essential for the growth and development of plants. Now, how it can be determined whether a particular element it is essential for the growth of plant. Now, there are three methods to detect this essential assessment or methods of assessment of essentiality we will be seeing now. So, there are three methods by which it can be determined. First is sand culture, second is solution culture, and third is aeroponics. Let us see each of these methods one by one. So first method is sand culture. You can see the picture here, a plant, a basil plant, it is planted in sand. So what is this method basically? Plants, they are grown in sand. The medium, growing medium is sand. So sand, it is washed thoroughly, it is cleaned, it is made sterile, and it is filled in earthen pots with drainage holes. And plants are, the experimental plant is planted in this medium. And this sand, it is supplied with the nutritional solution or with the solution containing the different elements which are required for the growth and development. And to detect whether if a particular element is required or not, that is deleted from that solution. And other remaining constituents are kept the same. And the plant is allowed to grow. In this method, the plants, they are usually planted in sand. The sand, it is washed thoroughly, it is made sterile and filled in earthen pots with drainage holes. The plant, the experimental plant is planted in this medium. The nutrient solution is supplied to the plant using a drip method or any other method to check whether the particular element is necessary for its growth or not. That particular element, it is deleted from the solution and the other elements, they are supplied in proper quantities. If the plant it is able to complete its life cycle, it is able to complete its vegetative and reproductive life cycle, then that element it is considered to be non-essential. And if the plant it develops a particular deficiency symptom or if it is not able to complete its life cycle because of that one missing element, then that element it is considered to be essential. Now this method it is quite simple, it is reliable, and it is very useful for the detection of macro elements. This method it is not useful for the assessment of micro elements. Now in this method, in place of sand, crushed quartz or even perlite can be used in place of sand, and no supporting medium is required in this method. Now let us see the next method, that is solution culture. Now in solution culture, also known as hydroponics, plants they are grown in a medium which is the nutrient containing water. or these plants, it is not cultivated in soil, but soilless culture or water culture, that is the name solution culture. In this method, nutrient containing water, it is supplied in tanks, and the plants are grown in this different types of nutrients which are required for the growth and development. They are continuously supplied in this drainage pipes or in these tanks, and the plants can be grown here. You can see in this figure. Now, this method, it is quite popular nowadays for the cultivation of different types of crops. Now, in this method also, same principle can be applied, that you can delete one element in the nutrient medium to check whether it is essential or not, and it can be tested on the same method. That is, if in the absence of a particular element, if the plant, it is able to complete its life cycle, then that element, it is considered to be non-essential. But if it, the plant, it is not, if the plant, it shows some deficiency symptoms and it is not able to complete its life cycle, then that element, it is considered to be non-essential. The next method for assessment of an essential element is solution culture. In this method, the plants, they are grown in nutrient containing solution. This method, it is also known as hydroponics, which is quite popular nowadays for the cultivation of various plants. In this method, the nutrient containing solution, it is flown in the containers or in the drainage system, drainage pipes, in which plants can be grown. You can see in this figure how the different plants, they are grown in the tanks containing the nutrient rich solution. In this method, the roots, they are in direct contact with the nutrient solution, and they absorb the nutrients from that water, nutrient rich water, and complete its life cycle and grow properly. In this method, air is circulated in this system so that the solution can pass and an aerobic environment is maintained here. In this method also, uh, whether a particular element is essential or not can be checked using the same principle. That is, the test element, it can be deleted from the nutrient solution, and the plant can be checked whether it is able to complete its life cycle or not. And thus it can be concluded whether a particular element, it is essential or non-essential. The third method, which is useful to assess the essentiality of an element, is aeroponics. In aeroponics, the plant material, it is the roots, they are kept in a chamber, and the nutrient containing solution, it is flown through the drainage pipes, and it is sprayed in the form of mist. That is the chamber. In the chamber, the roots, they are in contact with mist or the droplets of the nutrient rich material. In this method also, you can determine the essentiality of an element using the same principle. And in this method also, you can see how the roots, they are, an aerobic environment is maintained by the spraying of that liquid nutrient medium. Now let us see the role of minerals in plants. First major role of mineral, that is constituents of plant body. Now the minerals, they form important structural elements of plant body. For example, nitrogen, it is present in proteins, it is present in nucleic acids. Phosphorus is also present in nucleic acids. And also, if we see the other macro elements, for example, carbon, hydrogen, and oxygen, they are also important structural constituents of plant body. So that is the major role of minerals. That is, these minerals, they are important constituents of plant body, and they are important part of different structural elements of the plants. Second role is osmotic pressure of cells. Now different minerals, when they are absorbed by the plants from the soil, they can influence the osmotic potential of cell sap, and that osmotic potential, it is important for different processes of plants. For example, potassium, the element potassium, when present in cell, it increases the osmotic potential of cell sap, and it has a major role in opening and closing of stomata. So that is the second role of minerals, that is osmotic pressure of cells. Third is acidity and buffer action. Now, the elements, when they are absorbed from the soil, they affect the hydrogen ion concentration of cell sap or the protoplasm, and in turn, they affect the pH of the cell or pH of the plants. And that is also very useful in maintaining of the different buffer systems of the plants. For example, the phosphate buffer is the most uh, famous example. Next is catalytic effect. Now, different enzymes, they require cofactors or they require metal activators or which act as prosthetic groups, and they help in the overall functioning of the enzymes. So these different mineral elements, they also have catalytic effects, that is they act as prosthetic groups. Also they are present in the chlorophyll. For example, magnesium ion, it is present in chlorophyll, and the different metal ions, they are present in the enzymes which help in the different biochemical reactions. Also, if some elements, they can have toxic effects on plants. So these elements, when present in plants or if they are absorbed by the plants, they can have some toxic effects on plants. For example, lead, arsenic, mercury, these are some of the toxic elements. So the student assignment for today is write in brief about different types of minerals required for plant growth and describe their deficiency symptoms. You can submit the assignment on this mail ID. Now let us see the summary of today's session. Today we have discussed about mineral nutrition. What are the different elements required for the plant growth? What are the different classes of minerals? That is, we have seen the two major classes, macro elements and micro elements. We have seen the criteria of essentiality, why, how they are considered to be essential or non-essential. We have seen the three methods which determines whether a particular element is essential or not. We have seen sand culture, hydroponics, aeroponics, and these three methods, they can be used to determine whether a particular element is essential or not. And we have seen the different types and the role of mineral elements. Now, why the study of mineral elements, it is important? Because in a country like India, where agriculture is a major occupation of the people, mineral nutrition or the knowledge of mineral nutrition is very important, so that the farmers, they can cultivate the crops properly and they can apply proper fertilizers in appropriate amounts. So in order to achieve food security, we need to have proper knowledge of mineral nutrition. We should encourage more and more soil testing, so that a lot of money is not wasted in case of overuse of fertilizers or underuse of fertilizers. An appropriate quantity of fertilizers is used by the farmers. In the next lecture, we will be seeing how these different mineral elements are absorbed from the soil. We will see the different mechanisms by which these mineral salts are absorbed. And we will see assimilation, how these different mineral nutrients are assimilated. Thank you so much for your time, dear students, and we will meet next time. Thank you.